Good evening and welcome. I'm Cynthia Brandt. I'm CEO of the Lucille Packard Foundation for Children's Health, and I am telling you, we are going to have an incredible time tonight together. Now, I realize that's a big claim, especially for yet another online event that you're all going to. But I can make that claim that we're going to have an incredible time together for three reasons. First of all, because we are taking on a topic that is a big bet, a huge bet, one of the most audacious ideas I've ever heard, curing the incurable. The faculty, the superstar faculty that we're going to hear from won't settle for anything less than that. That's why we have the CDCM, the Center for Definitive and Curative Medicine, to bring together brilliant, audacious people with the resources of Stanford Medicine, Stanford University, and Packard Children's Hospital, and also the resources of this incredible community, all of you who support this work. Thank you. The second reason I can say that this is going to be an incredible evening is that you'll be moved. This will touch your heart. The faculty, yes, of course, you'll be emotional there too, but especially as you meet some of our patients and learn about their experiences, their ongoing complex experiences, which will remind you why we're all here, why we have this shared purpose. Now, the third reason that we're going to have an incredible time tonight is all of you. We have an incredible group of guests tonight. Now, I wish that we were all sitting down for dinner together tonight, getting to mingle over a meal and get to know each other, but instead, you have to participate online. So we'll have 2 question and answer sessions with faculty. In fact, you can look now and see the buttons on the left of your screen. So you'll be able to click there to get more information and to start entering your questions. In fact, I say jump to the front of the line, enter your questions now. Now, my final point is introducing the most incredible person in this incredible night. It's my pleasure, it's an honor to introduce Dr. Maria Grazia Roncarola, the director of the CDCM. She's a visionary, she's a trailblazer, a renowned scientist, a compassionate physician, and she's the proof. She has actually done it. Maria Grazia has found cures for childhood diseases that were considered incurable for millennia. She leads this team on a mission. She's a force of nature. Maria Grazia, over to you.
Thank you. Thank you, Cynthia, for the very kind introduction. And thanks to all of you for being here with us tonight. I know that there are many familiar faces on the other side of the screen, incredible supporter of our Center for Definitive and Curative Medicine. And I also know that there are new faces. And I want to thank all of you for taking the time to be with us this evening, despite your very busy agenda and calendar. I have to say that as an Italian, to have a virtual meeting is really a punishment. I like to shake hands with you. I like to hug. I like to kiss. I like to hear stories. But here we are, virtual. Hopefully, we will meet in person soon again. But even through a computer screen, I hope I can transfer to you the passion we have and the motivation we have to cure children with incurable diseases. This is the mission of the CDCM. And we want to cure these children with stem cell and gene therapies, which are a new class of drugs, what we call living drugs. They will transform medicine, and it's already transforming medicine. So these are drugs that have the potential to cure a child, a patient, not to treat it, to cure it. And also has the potential to be one-time intervention for the lifespan of the patient. So this is an incredible revolution for medicine and for physician scientists like me and all the faculty that have been used to prescribe pills, injection, sometimes surgery. Now we will prescribe cells. And the reason why we build the Center for Definitive and Curative Medicine is to close the gap between the lab and the patients, the bedside, and bring these living drugs to the patients and give to the patient new therapies, new opportunities, new cures. You may ask to yourself, why? Why do we need to do it at Stanford in an academic setting? Why industry cannot do it? Why biotech cannot do it? Well, you know, living drugs are a very peculiar type of drugs. They are very personalized. Most of the time are patient customized. And most of the time, industry, especially big pharma, are not particularly interested to develop this drug for children who unfortunately very often have the short side of the stick, and also especially for rare diseases. So we built from the ground the Center for Definitive and Curative Medicine in the past 5 years to bring these drugs to our patients, to the patient at the Lucile Packard Children's Hospital, and also to all the patients in need. So for those of you that have come to our dinners, you heard me talking for many years. We have to build the infrastructure. We have to build the infrastructure. Well, we did build the infrastructure. First of all, we recruited an outstanding team of faculty, really the best in the world. We built a laboratory for cell and gene medicine where we manufacture the drugs for our patient, the living cells. We have a fantastic clinical trial office that support us with the implementation of the trial to make sure that we follow the FDA rules and we are fully compliant. And last but not least, we had the incredible gift from the David Lucile Packard Foundation to build the 5th floor, where we have dedicated beds for these patients undergoing stem cell therapies. So thanks to this infrastructure and thanks to the know-how of our outstanding faculty, we have now 5 platforms. The platform of stem cell and transplantation, led by Alice Bertaina and Agnieszka Cekanovic. The platform of gene editing, led by Matt Porteous and Natalia Gomez Espinosa. You will hear more from Matt in a few minutes. We have the platform of engineering the immune system, which I lead together with Rosa Bacchetta. The platform of tissue regeneration with Tony Oro and Berta Cheng. And most recently, we added a new platform, the in vivo gene therapy, because we recruited one of the world leaders in vivo gene therapy, Fraser Wright. So the beauty of this platform is that in each platform, we can have many different diseases. And we can go from very rare diseases like severe combined immunodeficiency, what we call SCID, to more common diseases, unfortunately, as leukemia or sickle cell disease. So the uniqueness of these 5 platforms is that really a cross-fertilization and a tremendous synergy to cure many different diseases. So the example I always made in the past years is that I said we build a Ferrari. And now we need the gas to ride it, to riding. Now, I have to say to you that I'm incredibly proud to tell you that we got the gas. We got the support from all of you. We got the incredible support from the foundation and from the Children's Hospital, from Paul King. I'm incredibly grateful to Paul King for his continued support to the CDCM. And we got the support from all our staff. And in the past year, we have accomplished incredible goals. We have currently 5 therapies that are for the first time at the bedside, for the first time offered to our patients. And you will hear from the faculty for some of these therapies. But I can tell you that in the past year, a major accomplishment, we have been the first team in the world to replace chemotherapy with an antibody-based conditioning with a protein to transplant patients with stem cells from a healthy donor. This is a revolution, because not to give chemotherapy to our patients with genetic disease or leukemia, it will mean that they will not pay the price of the secondary tumor, of the sterility, of the many complications linked to that. We had the incredible accomplishment of getting our first gene correction trial approved by the FDA. I'm sure that Matt will come back to that. We have the opportunity to give to patients with leukemia one more tool to fight the leukemia. You will hear from Alicia Bertaina soon. And we have now more than 10 phase 1 trials from industry that knock at our door because they want to do the trial with us because of the unique know-how and infrastructure of the CDCM. So all the accomplishment of this year have a particular value because they have been during a terrible pandemic that hit very hard all of us. And it's only thanks to the dedication of our faculty and our staff that we could do it in this terrible 2020. I want to thank specifically Jennifer Corey, Alison Olsen-Speed, Niyar Bhatia, and the last incredible addition, Liana Lee, because without you, we would not have accomplished what we did accomplish. And what is next? Well, now we ride. Now we really are in the race. You understood that I am a fan of Formula Uno. So we did the qualification section and now we are competing. But we compete against ourselves. We compete to cure patients. We compete to give to every single child in our hospital and possibly worldwide the opportunity to live a normal life. And I want to share with you something very personal, and I hope that I can share it without becoming emotional. So a few days ago, I got an email from Shimon Slavin, a doctor from Tel Aviv. The title of the email was "Your Girl" in capital letter. I must admit that I opened that email with trepidation. I was afraid. But the first line of the letter said, "Your girl, the first patient treated with stem cell gene therapy, a young ADA kid girl from Palestine living in Jerusalem, is now turning 21 and is getting married." This made my day, and I can tell you, makes me even now very emotional. But this is why we are working so hard. This is why we need your help. And this is why I always say I'm impatient. People that work with me, they are wonderful. They know that I'm impatient because I know that there are children out there that we can help, thanks to our research and thanks to the CDCM. And this is an opportunity that we cannot miss. And we are all together in this. We need the help of everybody to achieve our mission. And with that, I know that I am already running over time, so I want to give the microphone to Matt Porteous and David Sanchez. Matt is the co-director of the Center for Definitive and Curative Medicine. He is a star in gene editing. We are incredibly fortunate to have him, and it is an honor for me to work with Matt and help him to accomplish his goal. Matt, take it from here.
Here we go. Much better. Being in the hospital isn't scary to me. Having a certain new problem isn't scary to me anymore because it's happened so many times. I don't know, my blood just does not like me very much, I guess.
It's often called the first molecular disease. It's caused by a single change in the DNA sequence. It's the letter A changed to a letter T.
That's it.
That's it.
That mutation causes a kink in the protein that prevents it from folding properly.
It becomes very stiff and it can't squeeze through. And you're not able to get red blood cells to the tissues where they can deliver oxygen. And if you block the ability of oxygen to get to those tissues won't work well and they'll get damaged.
David can go from crazy teenager, joking, jumping around, to a fetal position on his knees.
It's like pulsing. This hurts. You're having a sickle cell crisis.
We just didn't have the tools to make that single letter change in a precise fashion.
Even one letter.
Especially one letter. Before CRISPR, we were getting 1% to 2% correction. We're now up to 50% to 80% of the cells. This could really work. This could really cure the patient.
My greatest hope for patients like David and all patients with sickle cell disease is for them to never remember. We have the infrastructure with the 5th floor of the Lucile Packard Children's Hospital to treat these patients.
This is a first time that this type of therapy has ever been tried to directly correct a mutation. This is a really exciting moment because the concept of gene editing, the concept of one day being able to correct mutations that cause disease has been around for 40, 50, maybe even decades more than that. But we now have the technology at hand to allow us to implement what has only been drawn on a chalkboard before. It's incredibly exciting to be in the Center of Definitive and Curative Medicine, Stanford and Lucile Packard Children's Hospital, where we can take it all the way from the bench to patients.
Hello, everyone, and thank you for watching that clip from a recent documentary. I'll have to admit, that was the first time I've ever seen it, and I actually couldn't hear. So now I'm going to actually have to go back and watch it and listen at the same time. I've been far too embarrassed and mortified to think about watching myself on film. So really, David Sanchez is the star of the show. And I think this young man is incredibly eloquent, thoughtful, mature, and I think he described what it's like to live with the disease and what it's like to think that he might be cured of the disease. As Maria Grazia mentioned, we are at the cusp of being the first center ever to be able to take a patient's own stem cells, blood stem cells out of their body, correct the mutation that causes their disease, and give them back as part of an autologous bone marrow transplant. And there's no way that would have happened without the incredible infrastructure that was built. And I helped assist Maria Grazia in building the Center for Definitive and Curative Medicine that enabled us to bring discoveries at the bench, and you saw David looking at his own cells under the microscope, to therapies, first in human, curative, one-shot therapies for patients. We're incredibly excited that the FDA gave us approval last year to initiate the trial. We're incredibly excited that the institutional review at Stanford, which all these trials have to go through, was approved, and that we're on the cusp of being able to enroll and treat patients in the next few months. This would have not happened without grant support from the California Institute of Regenerative Medicine, but it also would not have happened without the generous philanthropic support we received to the CDCM and to my program in particular to enable this to happen. So thank you very, very much for that support. Finally, I'd just like to say that while sickle cell disease is an exciting first demonstration of genome editing to cure patients, it is only the first of many diseases we hope to cure. And while the CDCM was incredibly enabling to this program, it's going to be incredibly enabling to tens of programs over the next several years, thanks to the infrastructure that's been built, the generosity of the community around us, and the talented people we get to work with. Again, thank you very much.
Speaking for all of us in the audience, Matt, that is incredible. It's so inspiring, and it is really good to be part of this moment. on the verge of delivering a cure that will help so many people who've been suffering for so long. I'm honored to raise a glass. I hope everyone will join me to toast this incredible milestone along with all of the transformative work of the CDCM. Cheers to Matt, cheers to Maria Grazia and the team, and to amazing breakthroughs that will benefit generations of kids to come. Cheers.
Thank you all.
Thank you.
All. Thank you.
I'm now going to hand it back to Matt and Maria Grazia and an opportunity for everyone to ask your questions.
Okay, the first question is for Dr. Porteous. Does Matt's work in gene editing have the potential to be applicable to disease other than sickle cells? How would that work?
That's what's so exciting about the platform, the genome editing platform, is it can be applied to sickle cell disease, which is an extremely important disease, as you just heard about, but can be also applied in theory to every genetic disease that affects patients around the world. So the classes of diseases we are addressing next are the classes, other genetic diseases of the blood and immune system, because we can build on the sickle cell disease, what we've done for sickle cell disease for these other diseases. But when you think of diseases like hemophilia and cystic fibrosis, or even the very rarest of diseases, this platform is the platform that allows us to correct disease-causing mutations for any genetic disease. Now, each disease has its challenges, and I'm not saying that these will all happen tomorrow, but in theory, this can all happen.
Thank you, Matt. I see another question that's for me.
Okay.
Maria Grazia, what inspired you to be a scientist? Did you pick Stanford to launch the center? Why did you pick Stanford? Well, these are two very good questions. Well, I have to say that I've been incredibly fortunate in my life, professional life, and also personal, I have to say. I have two beautiful children, grown up. They would be offended when I call them children. But I've been incredibly fortunate because when I started my medical school, I was treating patients with SCID, and they were all dying. And now we can cure almost 100% of these children. I would say 99.9. So that's what inspired me to be a scientist. I could not live with the frustration of saying to a parent, your child is going to die. I cannot do anything for that. And what I said in my introduction, this is why we all work so hard. At the end of the day, of course, we care about recognition from our peers, but the most important thing is really to give an opportunity to children that otherwise would not have an opportunity to live or to have a normal life. Stanford is Stanford. When you think about the incredible critical mass of cutting-edge science in stem cell gene therapy, I cannot think about a better place to build a Center for Definitive and Curative Medicine. Thanks to Herb Weizmann and to many scientists, we have a beautiful stem cell institute, which is really unique. And is unique also the science, the breadth of the science that is ongoing there. So when I decided to come back to United States, of course I went to interview in many other places, but Herb Weizmann said to me, you have to come to Stanford to build a center for definitive and curative medicine. And I would like to add that the most convincing people to convince me to come were Matt Porteous and Tony Laura, that said, we need you. And here I am. And I never regretted that this is the right place to build such a unique center. Okay, Matt, are there any ethical questions about gene editing that need to be considered? We have all heard about the concept of designer babies. Are there potential applications of the technology that worries you?
Yeah, that's an excellent question and has been a topic of conversation for a number of years now. So What we're doing here at Stanford and at Packard Children's Hospital is what we call somatic cell editing, which means that we change the DNA, we correct mutations in the patient's own cells and their somatic cells and give them back. But when the patient passes away, which we hope will be in about 8 decades, those changes will die with the patient. The changes will not be passed along to future generations. But people have recognized that the power of gene editing might allow someone to modify the DNA, the DNA of a zygote or an early embryo, and as a way of making what you just mentioned, the designer baby. And I've been part of these conversations because I believe that that's not the right application of CRISPR-Cas9 genome editing. And that while there's a temptation to say that this is a way of making human beings better or preventing disease, I've now come to believe that crossing that boundary is not something we need to do to make the lives of patients better. That by enabling somatic cell gene editing, we can achieve everything that we could achieve by doing embryo editing or germline editing. Finally, I'll just say that what occurred in China, and I won't even call it research, was completely unacceptable, was outside the bounds of science, and is something that I wish I actually had met. As I said, I won't even call him a scientist anymore. I met him. He described to me what he was going to do. I forcefully told him that he should not do that, that it was unethical. But I wish, in retrospect, I had been more forceful and been stronger in stopping him from doing something that was really unacceptable.
Thank you, Matt, and thank you to all of you for the questions. I think we have to move on. We have an exciting program still ahead. It's my pleasure to introduce Rosa Bacchetta. Rosa will talk about a disease that we hope to cure very, very soon in the coming months with your help. And also we will have a video from Taylor, one of our patients with IPEX disease. Following that, we will hear from Rajni Agarwal and Alice Bertaina, how we will improve the life of our patients with leukemia. Rosa, take it from here, please.
As a doctor, most of my work is focused on understanding the mechanism of the diseases to find better treatment which are really targeted to the disease cause. This particular disease I'm working on, IPEX syndrome.
IPEX is an autoimmune disorder where your body is basically attacking itself, and it's attacking healthy tissue, healthy organs. So it really makes it hard to do certain activities in your daily life. Growing up with health struggles was definitely a challenge. At school, I felt like an outlier. I felt kind of like that weird kid that no one really understood.
He is really an example of how to cure the symptoms, to treat the symptoms is not sufficient, and we really should do more for this disease to avoid the quality of life impairment that can be associated for these patients and the family with them. My team has developed a process that is collecting defective cells of the patients and engineering them with the right healthy gene so that they become functional again in the right manner and they can be infused back to the patients. So it's a process with the autologous cells of the patients. We are optimistic that by the summer of 2021 we will receive the approval for this trial. The phase 1-2 trial is really near to begin. After that, the patients can be enrolled immediately, and we already have patients that would wish to have this treatment. IPEX is also very important because it really teaches us how we can treat autoimmune diseases such as inflammatory bowel diseases, rheumatoid arthritis, type 1 diabetes, and allergies. This treatment has the potential to really be helpful for a large number of people.
To know that someone is actually out there trying to find a cure for IPEX is pretty remarkable. It gives me hope for the future that maybe I can live a somewhat normal life.
To have the possibility to work on this innovative, high technological treatment is an incredible drive and is certainly a privilege that I would like to share with as many people as possible. Of course, with patients and their family, but also with young generation of doctors and scientists.
Hello, good evening. Welcome to the exciting evening. I'm Rajni Agarwal. I'm a clinician scientist and a stem cell transplant physician, and I treat children who have life-threatening blood and cancer disorders. As many of you might know, leukemia is one of the common childhood malignancies. And in many high-risk leukemia patients, chemotherapy just isn't enough, and we have to perform a stem cell transplant. And for that, we need to have fully matched donors, and many a times we don't have such donors available, and we have to resort to using mismatched donors for providing a chance of cure to these high-risk patients. Now, these transplants really bring a couple of unique problems of delaying the immune reconstitution, where the infections really predominate, graft versus host disease. And you know, the risk of leukemic recurrence always keeps us on our toes. What to do? And at that point, Maria Grazia came to our rescue. She has developed these unique T-regulatory type 1 cells. These cells were brought by her to CDCM for clinical trials. And what we learned about these cells, that they helped with immune reconstitution, they helped with decreasing the risk of graft versus host disease, and also preventing leukemic relapse. I have to say that I am the one fortunate person to be the principal investigator on the first clinical trial utilizing these cells. And we have treated 4 patients on this trial, and it is incredible that all these patients have tolerated the cells really well. They are safe, they're alive, they're leukemia free at this point. It is a great moment for all of us at CDCM. I am going to introduce you to Mark Torres. Now, Mark is a special young man. He was the very first patient enrolled on this clinical trial. He is now a thriving 11th grader, straight A student, and he's loving to play basketball. This young man has impressed me so much with his bravery, his resilience, and his kindness throughout the transplant process. And I am sure when Mark talks to you, you will love him as much as I do. Mark Torres.
Thank you, Rajni, for the kind words, and of course, for helping me on my battle against leukemia. I was a 13-year-old boy enjoying my summer vacation. Then suddenly I was having severe chest pains. I had a loss of appetite, and I was feeling extremely weak. Right away, my mom and I noticed something was wrong. After being taken to Stanford and a night full of testing, I was later diagnosed with leukemia. My mom took the diagnosis very hard, but I was ready to fight to be healthy again. Unfortunately, though, after further tests, it revealed that my leukemia was very rare and I needed a stem cell transplant. Eventually, I was able to meet my stem cell team, and they had given me a plan. Part of the plan included finding a donor. After searching for days, there wasn't a 100% match with a donor, which meant a mismatched donor was the only option. During preparation for the transplant, I enrolled in a new clinical trial which gave me a special type of T cells. These cells will help prevent graft versus host disease and help with engrafting. And of course, the course of transplant was very difficult. It consisted of 3 days of radiation, 3 times a day, and 2 days of chemotherapy. I spent many months in the hospital due to various different complications, but eventually things just kept getting better and better. Especially after I was discharged, it was nothing but improvements. Here I am 3 years later, being able to enjoy myself with no physical limitations. I'm looking forward to going to college and my future and working in healthcare. I am thrilled and proud to be part of this clinical trial and the future it holds. I can't wait to see how this trial can potentially save someone's life and change it for the better. Thank you for giving me the opportunity and allowing me to share my story.
Thank you. Thank you so much.
Thank you, Mark. Thank you so much for sharing with us your inspiring story. Good evening, everyone. My name is Alice Bertaina, and I am one of the other faculty within the CDCM. I'm also the chief of the stem cell transplant program. But first of all, I am a pediatrician. And like everybody else in the Center for Definitive and Curative Medicine, I have a quite ambitious goal, because really what we want to do here is cure as many children as we can. And let me thank first Rajni for her leadership in this field and for the collaboration for trying to treat these children with acute leukemias. Thank you, Rajni. This story of Mark is one of many stories that really are our driver every day. And when I started my career as a physician scientist, was really the story of a patient like Mark who couldn't identify a completely matched donor. And for me, as a young scientist, that was something really unacceptable. How can we not give the chance to a child to be treated from the leukemia just because he doesn't have a common DNA that we can find somewhere in the world? And so this is where really my research started, because my goal was to identify a donor for everybody in the need of a transplant. And I can say right now, I'm pretty proud to say that we are there. We can basically offer a safe transplant to everybody in the need. And this is why we were able to play a little bit with the cells and try to select and eliminate from the transplant the bad cells that are the one that can be responsible of graft versus host disease. And in this way, we can use one of the two parents, even if they are just a 50% match with the recipient, and avoid that develop a severe graft versus host disease, which can be a lethal complication. But unfortunately, this plan is not perfect. And every time we have to eliminate something from the transplant, we are gaining some point on one side and we are losing something on the other side. And that's where the CDCM becomes really the right place where to run these clinical trials. Because within the CDCM, my expertise on alpha-beta T-cell depleted haploidentical transplant, this is how we call in technical terms when we do this transplant from one of the two parents, get married with the regulatory type 1 cells of Maria Grazia and Rajni. And so we now are launching a new clinical trials where we are combining these two therapies. Why are we doing that? Well, because in this way, we really can offer a transplant to everybody, but we need to have more of these cells to fight the infection, like Rajni was saying, and especially to reduce the risk of leukemia relapse. And the TLO10 cells, which are the specific cells manufactured thanks to Maria Grazia science, are able to provide this effect, are able to fight the infection, to promote protection against the leukemia relapse without creating graft versus host disease. So they are the ideal, optimal tool for really curing our patients. We are doing that. This trial has been approved from the FDA. We are open for enrolling patients, and we actually have enrolled the first patient who has been already transplanted and today is on day plus 13 after the transplant and fully engrafted. And we are aiming to infuse the first dose of these cells in 2 weeks. but I need your help. I need your help because just with the generous contribution of the philanthropy, we can extend this treatment to as many children as we can. We need to get wherever we can. We need to treat patients who can come from all over the country. And for doing that, we need your help. We are a team within the CDCM. Maria Grazia, Tony, and Matt had the vision of building this inspiring organization, building the infrastructure, as Maria Grazia was telling before, and putting together all this faculty and their science. But without your support, we cannot really do anything. Thank you so much for listening to us. Thank you so much for participating in this event. I really hope we can very soon get together in person and showing you even more what are the progresses of our research and our clinical trials. But it's a pleasure for me to be here and to share, at least virtually, with you where we are and where we are going. Thanks again.
What inspires me are the patients and the ability to use whatever scientific insights to make their lives better. If you just see one kid with epidermolysis bullosa, you want to do everything you can to help them. These are rare diseases that we're treating, but they're useful for a wide variety of other more common diseases as well. For example, we can genetically correct the skin and then manufacture skin at large quantities to put back on patients. But that same type of manufacturing can be done for wounds, burns, diabetic wound healing, injured soldiers. There's a broad range of applications that the technology can apply to once it's into the clinic. The main thing to take away today is hope and the potential for the Center for Definitive and Curative Medicine for the future. We have shown you that both in the scientists that are part of the center as well as the families that we interact with, that there's a very strong bond between us. We are developing the therapies and working with the patients to really accelerate these therapies for them as one-step cures. We have over 24 types of therapies that are coming through our pipeline. There's so much more to do. There's so many more patients to cure, so many more diseases to attack, and we have the ability to do that with your help.
Good evening again. I hope that you really enjoyed to hear about our patients and our stories, and that you share with me the excitement that I feel, thinking that this is just the beginning of our adventure, and that we will have in the next 2-3 years too many therapies to describe in 1 hour virtual event. So I would like to open the floor for any question you have for me or for the faculty. We are all here to answer to your questions, and we are looking forward to that. So I see in the chat that the first question is for me. It says, Maria Grazia, how many current trials are ongoing and for which condition? Now, I think that if I have to list all the conditions, I'm sure I will forget few, but I can tell you that we have currently 18 trials ongoing, first in human, first in children. The trials that really stem for our discoveries are the trial with the T regulatory type 1 cells that Alice and Rajni described, the trial with the antibody conditioning, which is based on the work of Herr Weissman, Agnieszka Czekanowicz, and Judy Shizuro to replace chemotherapy. What we heard from, to replace chemotherapy for SCID patients, but also for patients with Fanconi anemia. Actually, I'm very excited that we just got an approval from the FDA to use this antibody conditioning combined with the technology that Alice was describing in the haploidentical transplant for Fanconi anemia. We have the gene editing trial of MAT that is open. We have trial in a rare form of anemia like PKD, and we have trials close to to be ready for metabolic diseases and neurodegenerative diseases. Actually, we already have one trial open where we engineer the stem cells of the patient to deliver the enzyme which is missing. So it's a variety of diseases and conditions. And this brings me back to what I said before, that with the same platform, the same technology, we can address many diseases. For example, with the engineering the immune system, we heard Rosa speaking about IPEX, but we are also thinking to engineer the immune system with the type 1 regulatory cells you heard from Alice. We will engineer the immune system by giving another subset of cells, the gamma delta cells that Alice is studying. We are building CAR-T thanks to the gene editing platform of Matt Porteous. So one platform can serve many diseases, from rare diseases to leukemia, which is really the strength of the CDCM, which is allowed this type of synergy across fertilization. Another question is, do you anticipate some of the therapy that were described tonight becoming standard of care for many diseases? If so, will that happen with our lifetime, and what will it take to get there? Well, this is a very important question. Of course, we are an academic institution, and our goal is to do cutting-edge breakthrough work. So to open the field for new therapies. But of course, the purpose is to have these therapies become drugs on the market, available for every patient in need. In my professional life, I was lucky enough to develop 2 therapies that reached the market. The first one was Trinvalis, and the most recent is Limlendi for metachromatic leukodystrophy, a very serious and severe neurodegenerative diseases. What I can say by experience is that the first time it takes forever, to develop Trinvalis, which is the drug that the Palestinian girl received. It took more than 15 years. But then the second one was much faster. And now it's going more and more, accelerating, and because we facilitate with our infrastructure. So I anticipate that the gene therapy, the gene editing, the gene correction of MAT will reach the market much sooner than what we saw with Trinvalis or other drugs. But maybe I'm curious to hear from Matt if he agrees with me. I would anticipate 5-7 years, which is very aggressive.
Yeah. 5 to 7 years from giving the first patient a new medicine to approval. It would be a very rapid timeline, but I agree that what we have learned from Strimvelis and the gene therapy for metachromatic leukodystrophy will help us accelerate so that we can go faster. And not only do we learn about how to accelerate these therapies, the regulators who approve these therapies also learn. And so we can, it is really a joint effort. And while as much as sometimes we all mutter under our breath about the FDA, the reality is in the United States, the FDA is a true partner with us because they recognize the serious nature of these incurable diseases and the desperate need these patients have. So perhaps 7 years. I'm not sure we can do it in 5, but I think we can get it in 7. But again, we're not going to wait 7 years before we do the next gene editing clinical trial. In fact, we expect that it'll be a pyramid, that we do 1 gene editing trial in 2021, we do 2 or 3 in 2022, we do 5 to 7 in 2023, and it will just be an exponential growth. And that, yes, I think within our lifetime, these are going to become standard of care therapies for many of these diseases.
Thank you, Matt. There is a question for Rosa. Is the gene therapy that you're developing for IPEX applicable to other autoimmune diseases such as type 1 diabetes or rheumatoid arthritis or even atopic dermatitis?
Yes, thank you for the question. This is very important, and is also my response is also very much related to what Matt just said, that before this LV FOXP3 product that we have for IPEX will reach the market, we will already test it in different diseases. And there are all these diseases that in the question have been mentioned, like IBD and type 1 diabetes and so on, are really due to lack of T regulatory cells and will benefit from this cell product.
Thank you. A question for Alice. Alice, since your technology allows to treat every patient, can we treat all the patients with many different diseases? What is the bottleneck?
Yes, absolutely. Thank you very much for this question. So, as we like to call it in the CDCM, this strategy of graft manipulation is a platform. It's a platform because we can apply it not only to acute leukemias, but also to children with non-malignant disorders, to children with thalassemia, to Fanconi anemia, like Maria Grazia mentioned, sickle cell disease, if they are not eligible for the genome editing trial of MET, and many others. And I can tell you, spoil some news for the future. That's we are going to have to keep it for next year. But we are opening a program for using this approach of stem cell transplant to promote tolerance in patients who receive a solid organ transplantation. What does that mean? It means that patients who do not have hematological disorders, but are unlucky enough to have some kidney disease or liver disease and are in the need of a solid organ transplant, can now receive before a stem cell transplant to avoid the need of a lifelong immune suppression, which is the major complication of this condition. And I can tell you that we are hardly working on that, and we will really have good news soon.
Thank you, Alice. A question for Matt. What is involved with the sickle cell treatment? Is there chemotherapy involved? What age patient will be eligible to participate in the trial? How long will the treatment take?
Those are very excellent and very insightful questions. Thank you for asking. So the process it involves first giving the patient a drug to push their stem cells out of their bone marrow into the bloodstream. And when they're in the bloodstream, we can do a process called pheresis to pull the stem cells out of the body without having to take them to the operating room. Once the stem cells come out of the body, they go to our special facility, which Maria Grazia mentioned, the Laboratory for Cell and Gene Medicine. Well, they'll undergo a 5-day manufacturing process to correct the mutation that causes the disease. Following the manufacturing process, the cell product, which is will be hundreds of millions of cells, will go through a series of tests to make sure that it is sterile and we have done what we thought we have done. And if it passes all of these tests, which we expect it will, then the patient will come back to the 5th floor of the Children's Hospital. And yes, in 2021, the patient will have to receive chemotherapy to get rid of all of the residual hematopoietic stem cells, blood stem cells in the bone marrow, that would give rise to sickle cell disease. And so not only are we getting rid of the cells that cause sickle cell disease, we're making space for our gene-edited cell product to take up residence in the bone marrow to make blood for the rest of their life. So yes, but what we hope is that because at Stanford we're developing ways of creating space in the bone marrow without having to use chemotherapy, that one day the peanut butter of gene editing and the chocolate of non-chemotherapy condition will come together and form a delicious dessert that we will use to treat people all over the world. So that's one of the really exciting things about being at Stanford and being part of the CDCM, is to see these different programs, you can see eventually how they're going to come together and really define what people have described as the holy grail of curative therapy for patients. I forget the last part of the question as I was talking, but
no, I think that's how long the treatment will take.
Yeah, sorry. So the treatment in the hospital is right now like a standard stem cell transplant, where the patient is likely to be in the hospital for somewhere between 1 and 2 months because their immune system and their bone marrow hasn't recovered to be healthy enough to be outside of the protective environment of the hospital. The reality is that these patients, we will follow them the rest of their lives and monitor them very closely for the effectiveness of the therapy and to make sure that some unintended side effect doesn't occur. So they're going to be our patients forever, if they'll have us.
So there is a last question to which I can give a very quick answer. The question is, 18 trial seems a lot. Can you take more? And the answer is yes. If you continue to support us, if we continue to have the support of the Lucile Packard Children's Hospital, we can take more. We also have to say that our goal is to prove the efficacy of our new therapies and then really make it available for many other institutions. So what we develop is unique at Stanford, is Stanford only, but we don't want to keep it for Stanford. We want to make it available for every patient. And with that, I want to thank all of you for joining us. There are 4 breakout sections. I hope that you will be able to join one of those, and it would be wonderful to see you and to drink another glass with you. And with that, thank you. We are very grateful for your support. It means a lot for us. And we will continue to work to change the life of our patients. Cynthia, I give it to you.
Thank you, Maria Grazia. That is so incredible. I think I speak for everyone. We're inspired, we're moved, we're motivated to help. I especially want to thank David, Taylor, and Mark. You have touched my heart and all of ours. We're with you 100%. Thanks again for joining us this evening, and cheers.